Thank you for the invitation from the World Congress of Psychiatric Genetics 2021 conference. Appreciated all the supports from the conference organizers. My name is Zheng Jie. I'm a research fellow from MRC Integrative Epidemiology Unit at University of Bristol. Today, I'm going to present how to examine causality using Mendelian randomization. Mendelian randomization is Mother Nature's randomized control trial. Segregation of alleles at mitosis mimics the random allocation of individuals to the drug and placebo arms of a randomized control trial. If the number of disease events is lower in the drug arm, we conclude that the drug by its protein target was effective for reducing disease risk. Similarly, if we see fewer disease cases in the genotype AA subgroup, we conclude that the genotype by its protein target reduces disease risk. A major advantage of MR study is that they are much cheaper and safer to run than randomized control trials. How does the method actually work? In Mendelian randomization, we use genetic polymorphism or SNPs to proxy for the protein or other exposure of interest. We have two effect estimates: the effect of the SNP on the protein in a protein GWAS and the effect of the SNP on disease in a disease GWAS. A GWAS, or called genome-wide association study, measures the association between hundreds of thousands to millions of SNPs across the genome with a trait of interest, typically in very large sample collections. Thousands of such studies have been published in the past 10 years. In Mendelian randomization, we combine the SNP protein and the SNP disease effect estimate to infer the effect of the protein on disease. In the example, the protein level decreased by 0.5 units per G allele in the protein GWAS, and disease risk increased by 42% per G allele in the disease GWAS. Using the ratio method, we take the ratio between the two estimates to infer the effect of the protein on disease. The latter is the odds ratio of disease per unit increase in the protein and can be interpreted as a 50% lower disease risk for each unit decrease in the protein. Causal inferences using Mendelian randomization require the following assumption to be true. First, the SNP truly affect the protein. Second, the SNP affect disease only through the protein. And third, the SNP is not associated with confounders. MR can be used to rapidly evaluate the potential downstream consequence of pharmaceutical interventions, which including first, prediction of on-target beneficial effect, second, predict of on-target side effects, and third, opportunities for drug repurposing. To support large-scale causal inference using Mendelian randomization, we developed the MR-based platform and the two-sample MR package. The MR-based platform is both a database of GWAS summary statistics and an analytical platform for Mendelian randomization. As an example of applying the MR-based platform, we studied the causal relationship between protein and disease. Proteins are direct targets for most of the drugs and therefore have high priority for drug development. Using the MR-based platform, we are now able to estimate the putative causal effects of thousands of proteins on hundreds of human diseases and risk factors using Mendelian randomization. This large-scale Mendelian randomization study identified over 200 robust protein disease associations, which covered a wide range of disease areas, including psychiatric diseases, cardiometabolic diseases, autoimmune diseases, and cancers. This analysis also validated 8 approved drugs to the same indication being investigated in existing clinical trials. Moreover, we systematically compare the MR estimates with clinical trial effects. We found that it is considerably more likely to predict drug trial success if a protein disease pair show MR and colocalization evidence. To improve reproducibility, we made our protein disease association results available via the EpigraphDB platform. The EpigraphDB platform is an analytical platform and database that aims to support data mining in epidemiology. Within the platform, we are able to query the protein we are interested in, the outcome we are interested in, or even the SNP we are interested in. And the platform will return the graphical results and the MR results in a table. In addition, we recently upgraded the MR-based database into a centralized GWAS summary database. The new database is now launched as the IEU Open GWAS platform. Till now, the new platform provides summary stats for over 40,000 GWAS traits. To further extend the usage of GWAS summary statistics, we developed this ecosystem within the IEU Open GWAS project. The ecosystem including both database and analytical tools, which could be accessed via a few R packages, such as IEU GWAS R and IEU GWAS Glue. These R packages could be used to support all types of genetic analysis using GWAS summary statistics. This includes LD score regression, fine mapping, genetic colocalization. Hope the ecosystem could make your research life easier. This is the end of my presentation about exam causality using Mendelian randomization. Thank you for your attention.